Be careful of what you say. Only smart comments, please. Nothing else. So today I'm going to tell you about part of the image project run. So this is the project 2 bit, which is about predicting gene functions using phylogenetics. This presentation, it's just a part of my qualifying exam. So I kind of like chop it and like clean it a little bit. So if you see something odd, don't worry, it's just because I may forgot to remove that part. Okay. So yeah, that's not supposed to be, that's the end. So let me actually go to the beginning. Okay. So this is joint work with Professor Thomas, Paul Thomas, former John Wyoming, Duncan Thomas, and John Morrison. Okay. So genes. Let me tell you a thing or two from let me give you like a crash course on biology from an economist's point of view. Okay, it's kind of odd, but I'll do my best. If I say something out of place, just let me know. I'm an economist. Please be nice to me. Economist aspiring to be a biostatistician, so I'm doing my best. So genes encode the synthesis of genetic products that ultimately are related to particular aspects of life. And so, for example, we do have some genes that are associated with the molecular function. Okay? For example, you can have a gene that is associated with the active transport in and out of the cell. There'll be a gene with code term 005-15. I'll tell you more about all terms in a bit more. Or it could also be associated with some cellular component, like a part of the cell itself. Okay, so you might encode something about the way that, I don't know, the structure of the genes of the cell itself. So how, I don't know, it could be mitochondria, for example. This is another gene that is associated. This an annotation that is related to the mitochondria component. Or it could be something bigger, like, for example, associated with the heart contraction. So it might generate I don't know, some molecule or something that is related to that biological process itself. Okay, so genes encode all this sort of information. Okay? And for that, we actually have a huge project, as I guess that most of you know, which is the Gene Ontology project, right? Which is an international collaboration in which we're essentially trying to create the biggest library body of knowledge about biology in general, not only human, but in general, right? The GOAT project right now, as today, has around 44,700 validated terms, as the ones that I just mentioned you. It contains about 7 million annotations, okay? In the sense that you have like pairs of genes or other kind of molecular parts of the genome associated with an annotation on about 4,500 species. Of these 7 million annotation, 500,000 are in human genes. Okay, but that doesn't mean that we have 500,000 genes, human genes. But actually, I realized about this not long ago that humans, we have around 20,000 genes. That's like the latest count, and roughly half of that have some sort of annotation, which means that we know barely. We just know a little bit of ourselves, right? And furthermore, if you're thinking about across genes, across multiple species which are about like 1.7 million, we only know something about 10% of them. So there's a lot of things that we don't know yet that we need to investigate. Okay, all of this is information that I took from the Panther project, which is part of the Gene Ontology and the Gene Ontology website. So GOAT term. So this is an example of a GOAT term. Okay, so for example, here we have the GOAT term, that code number. It's associated again with heart contraction. It's part of a biological process. And it gives you some information about like cell names and some other IDs and a definition, a clear definition. Okay. What I found interesting about this function in particular is that, for example, this function is known to be associated with a gene of each one of these 4 species. Right?
Sorry, which function? Heart contraction in general, or?
With the annotation heart contraction. So we know that they have a gene that has this annotation, each 4 of these species. And the thing is that what it's more interesting even is that, so the numbers here, this is just the code of the Panther project. I'll tell you more about it in the next slides. Of these species, actually, these two species are in the same evolutionary tree. So besides of sharing some information about like the gene annotations, we also know that at some point in evolution, these two species like share something. Okay.
I mean, isn't it?
Everybody, yeah, but the thing is that they have part of the same family. So you know, as I'll show you in a few minutes, we actually organize species, genes, in different as families, as group of families, and these two in particular are part of the same family from the biological point of view. Okay, and here it is. And so everything is connected in regarding a phylogenetic tree. So this is how we organize this information about how species and actually genes are, let's say, move along evolution. And here's an example of a big one. So this is like I would say it's not the most the earliest one, but here's a phylogenetic tree of all living things, right? But it's based on RNA comparisons. And you have in overall, you see like we have three big groups. You have bacteria, archaea, and eukaryotes, right? So this is what I'm talking about usually when I think about trees, about like families. But this is like the overall view. We will see some that in general we're actually thinking about smaller groups, which takes us to the Panther classification system. Okay, so in the Panther, which is part of the Go project, we have information about evolutionary structure of about 1.7 million genes. So instead of looking actually at species, we're looking at genes of species. Okay, so in the past, it used to be the case that for us to build these trees, we would look at features of the species, of the animals. Like we would say, okay, does it have fur? Yes, no. Does it have wing? Yes, no. But now, we actually, instead of that, we actually look at RNA or gene sequences. So we actually compare those to build these trees. So instead of putting the species on the tip of these trees, we actually put genes. You can ultimately, still build an evolutionary tree based on species only, but the way that we analyze it these days is actually like this, at the gene level. And in Panther, again, we have 1.7 million genes organized this way. These are grouped in roughly 15,000 families. Okay, so as I would say, so when I have a picture of the lizard and the fish in there, actually they too are belonging in the same group in one of these families. We are saying that they are, compared to other genes, they are closer connected in terms of evolution. And the nice thing, again, of a family is that it can host multiple species. So for example, to say something, we may have a family that has genes of human and some other species that, I don't know, like the fly, or I don't know, other weird species, other weird connections that we wouldn't guess, but we have them there. And here's how they look like. This is an example of a simulated phylogenetic tree with some annotations. Here we observe the tree. Each dot in the tree represents an ancestral node. So we have, for example, we have here the root node, like allegedly the first of all. And then at the end of the tree, we have the tips represent modern genes that we know of, right? And furthermore, besides of having this information, we also have in this simulated tree, we have two sets of annotations. So we have function 0 and function 1, which could be mapped to like gene ontology functions. And in this case, if it has if it's colored blue, it means that it has the function. Red means it doesn't have the function. And white means actually we don't know whether it has the function or not. This tree would be really nice to have if all trees were like this, but unfortunately, most of the data that we have on Panther is, I mean, in the gene ontology is actually very scattered. So it's most of it is colored white. So we don't know much about it. Another interesting thing, important thing to mention here is that another thing that we know, we actually infer from this from the evolutionary process is that we identify some special types of different types of internal nodes of ancestors. So, for example, here we are saying that this node in particular is a duplication event, okay? And the other ones, the gray ones, are speciation events. Why is that important? Because that would give us a hint of whether a new function was developed or there, or actually another one was lost. Which takes me to the next part, which is talking about speciation versus duplication.
Versus what? I couldn't.
Speciation versus duplication events.
Mutation.
duplication events. And let me take a brief pause here. So how we're going, is it too fast, too slow? Are the biologists okay with the way I'm explaining things here? Makes sense? Kind of? Okay. Johnson is okay. So. Yeah, if you say it's fine, I'm cool with that. So. Okay, so we do have, as I was saying, through evolution, we identify two different types of events. We have speciation events and duplication events. Speciation events essentially means that some external factor, something that is not of the gene itself, made two species to actually like split. There's a famous experiment in which by Dodd, in which I think it's she isolated a set of fruit flies of the same family, like exactly the same type of fruit fly. She isolated it and put it in different environments in which one of them was fed with maltose making, and the other one was starch making. So these two sets of fly were having different sets of food, okay, kinds of food. And she looked at how these flies evolved through developed through a whole year. I think it was exactly 1 year, and starting to see what happens if they develop any difference. And at the end of the year, then she mixed these two sets of species and asked them to actually put them together to see how they would mate. Okay, and when they compare the mating preferences, actually they identify that they develop differences to the point that they tend to significantly choose individuals who were part of their same group was more likely. Okay, this is an speciation. And so, for example, you have another thing that I don't know. Something happens ABC, and you have a single same species, and then I don't know. There's an earthquake and splits the earth, so you have they split themselves. Now you have a speciation event. Okay, so it's an external thing that may have no effect in the DNA, but it either way makes some differences ultimately. Does it make sense? But then we have duplication event, and this is directly to the DNA. Okay, so for example, we may have again, imagine the fruit fly again, but we have a gene. Okay, so we have a gene with 4 different functions, but then for ABC reason, this gene gets duplicated in its DNA. Okay, so either of 3 things will happen. Either either you have a sub-functionalization, which means that they specialize into a single set of functions. So for example, in this case, the first gene got this and that, like the red and orange function, and then the second gene got the blue and the green function. You may have, for example, another type of event, which is neo-functionalization, which means that instead of keeping the same function, they actually develop a new set of functions, the second gene. Or divergence, which means that essentially the gene is just lost and nothing happens. Okay? Duplication events are where biologists think that it's most likely to actually happen to have a new set of functional loose functions in a gene. So being able to identify which nodes, which ancestors in the phylogenetic tree are of type of duplication event is very important for us for this reason, right? Because we will inform whatever kind of model we're building to say whether a new function happened there or not. Make sense? Yes? No? Okay. With this, since we have these trees and we have the species, like the annotations on the Go project, actually we can use both evolutionary trees and observe annotations to inform a model prediction model for genetic annotations. Okay, so in other words, instead of staring at the genetic functions by themselves, we can actually grab all the information that we have accumulated from the evolutionary relations and try to tie these two things together to see how can we use whatever we know from these species to infer how likely it is that this other species has or doesn't have a function. Right? That's actually the punchline. That's the whole idea of the model. Okay, so this is a brief description of the model that we are thinking of. This is actually Jonathan's idea. So here we have a tree, right? An evolutionary tree, in which the leaf are the tip are the genes that we observe, right? Here we have a root node, which is in blue, And then in green, we have some internal nodes, okay? So in this model, we first imagine that there's a probability that describes the gain of a function, a spontaneous gain of a function, saying that this root node actually started with a function amount. So we call that pi. Then at the interior nodes, we imagine this is a Markov process in the sense that we have the chance that one of these offsprings, one of the offering of this parent node, in this case, this is the root, gains a function. It only depends on whether the parent has a function or not. And in particular, in the type of event that we're observing, the type. For example, if it is an speciation event, then the likelihood that this node has a function, given that this parent has a function, is described as pi naught. Otherwise, pi naught, pi t naught, or otherwise it's pi t1. This is the case that, for example, from this is a functional gain. I mean, not is one functional array. So from not having a function to having a function, and then the probability of losing a function. This is the parent has a function, then the offspring does not have a function. Okay, so these are the parameters that we're trying to capture, and this happens throughout the tree, at all interior nodes. Okay, and finally, another pair of parameters that we are adding to the model is we are trying to control for human error. In other words, we're trying to say see if, for example, a 0 actually was labeled as a 1. So we're giving that a parameter as well, which is the probability of mislabeling. Okay, so again, to recap, we have the root node that may or may not have a function. So with probability P will have a function, then it will start climbing down this Markov process, in each step, the offspring of the parent will have the function, will either preserve the function, lose the function, or gain a function as a combination of these probabilities conditional on its parent state until we reach to the leaf nodes. And once we reach to the leaf nodes, we also are putting a probability that actually the researchers may have mislabeled this probability. Mislabeled this annotation, saying that, okay, so actually it was a 1, but they label it as a 0. Okay? We implemented this model using Felsenstein pruning algorithm, which allows us to calculate likelihoods on trees in a linear complexity in the R package A5. Okay.
Which is a package I wrote.
Yes, which is a package I wrote. Okay, so let me give you some examples of. Also, this R package allows you to simulate and visualize this process of generating new trees and then annotated them according to this Markov process. It's written in C, so which uses the pruner template library, which is another library that I wrote for the project. If you are interested about using the Felsenstein pruning algorithm, it's written in C. I have it as a standalone library. It uses metaprogramming, so meaning that we can specify different formulas, including pooled data models, which means that instead of looking at a single tree, we can actually grab a group of annotated trees and pull them all together in a single model to do the parameter estimates. We can estimate parameters using either maximum likelihood, maximum a posteriori, which is just MLE with some prior information, and MCMC. And the later is actually done using the FMCMC package, which is already on CRAN, that allows us to implement automatic stop via convergence check, out of the box parallel computing, using multiple chains, and user-defined kernels. In the case of our model, when we fit this MCMC model, we're using adaptive MCMC, implemented as implemented in this package.
Which is another of your contributions.
Yes. Okay, so just let me jump right away into some results that I have. Okay, so this is how ultimately the model looks like. So again, remember that we have the tree, we have the teeth in which we have annotations. Most of the time we don't have much information, but either way, so what we did is that we took 141 trees, okay, that we got from the Panther data set, okay, which contain around 7,000 0-1s scattered across these 141 trees, and we pull them together to estimate this joint model. Okay, so what we have here is the parameter estimates, which happen to be also probabilities, and that's one of the neat things of the model. So instead of trying to use like exponential or something else to interpret the results, we can just look directly to the parameter estimates, which will tell us what's the probability of observing this or that. So let me start by saying the first observation here. So first, we fitted the model twice, okay? Once using MCMC with a uniform prior, and then using MCMC with a beta prior. And the first thing that you should be able to see is that the parameter estimates are not that different, which means that the results are mostly driven by the beta, which is pretty neat. The first time that I got this, I actually was kind of concerned because I was thinking, okay, maybe the prior got stuck somewhere in the program, and that's why it's stuck there. But actually no, so it's very consistent, and it makes sense because again, so we are using a lot of trees to pull this. So the second thing is that we actually observe what we believe is biological meaningful results. So if you remember from what I was describing early on, when we think about duplication events versus speciation events, we will put as biologists more higher chance of functional gains and loss at duplication events versus speciation events. And what the parameter estimates is telling us, what the model is telling us is actually exactly that. So the probability of gaining a function at a duplication event is near to 97%. And the probability of losing a function at duplication events is pretty close to 50%. So that goes in line with what was predicted by theory. But then if you look at speciation events, the chances of observing any change are very low. That's very nice.
You're probably going to say this later, but it's worth clarifying for everybody else at this point that for the time being, the gain and loss functions events are independent.
Right.
And we want to ultimately deal with the possibility that one branch will gain a function while at the same time the other branch loses.
Yeah,
that function.
that's a very good point. So actually, let me go back just to look at the tree again. So what Duncan is saying is very important. So for example, in another thing in biology, so every time that we observe a functional gain, it usually happens in one of the offsprings, only one of the branches. Actually, if we observe a change at all, either a loss or gain of function, it will happen in only one of them. So for example, either this or that will gain or lose a function. But our model right now, as the way it's specified, it's not allowing for that. It's just saying that functional gains or losses are independent between offsprings, which doesn't make much sense. But actually, we do have something. I will point you to something that we're thinking about how to correct for that in a bit more. Okay, and a final point before like getting out of this slide is that a couple of other things. At first, also if we look at the accuracy of our model, we actually use D1R AUC to see how accurate were our predictions. If we look at the median, and the mean, it's between 70 and 80, the AUCs, which is in line with what we have currently in state-of-the-art model. So one model that we're using to compare our results is a model that is written by a group at Berkeley, which is called Sifter. I don't know how many of you have heard of it. But the thing is that they have this huge machinery that it's, in theory, it works great, but it has some issues regarding computational efficiency. And the AUCs that they have reported in some of their simulation studies, actually some of their studies, is that it's around 70, 75. So we are in line with them. But here comes like the thing that I like the most about this is that for us to estimate these 141 full function trees with 7,000 annotation, it took about 5 minutes each. In Sifter, I don't know if that's not feasible. And I do have, I think I didn't put it here, but if you try to do this on Sifter as something that is not a completely fair comparison, but if you try to estimate a model that has this many functions, it will take like about 67 years, according to their website. They have a thing to compute how much time it will take you. But again, so it's not a direct comparison, but the thing is that for us being able to obtain this very nice accuracy level compared to what they get in just 5 minutes, it's remarkable. Okay, so here prediction. So here we have how does our model do when we have a fully annotated tree? So here, this is the true set of annotations, the first column. The second column is the predicted annotations of our model. As you can see, we're actually very, very close. The only problem here is that the points in which you observe this like more clear, which means that the prediction is, if the model is not very certain about the type of annotation, is because we have either a speciation or a duplication event at these points. And because of that, since the annotations of the neighbors here differ, the model is a bit confused about how to annotate these leaves.
So what did you start with?
What did it start with?
Yeah, what data did it start with to generate
The annotations?
It has a data set.
That's a very good question. So the way that the prediction works here is that we predict the ith observation conditional upon the other observations, whatever information we have. So when we have like fully annotated trees in a leave one out context in which we, for example, if we want to annotate this one.
Also leave one out.
Yes. If we want to annotate this individual here, okay, so we take them out by using the information of the entire tree to annotate, and because of that, we're able to get like these very nice results.
Okay.
When we have, for example, like more scattered annotations, this is how actually like annotations on the Panther and the Go project look like. So we have like mostly zeros. We don't know much about what's going on around. We do have some ones, mostly ones, and a few zeros, like a few not annotated leaves. But if you take a look here, for example, this is I took the parameter estimates for the pool data model, okay, and I used it to make predictions on top of this tree, which was not included in our original model. So when we are checking here out of sample prediction, how can we do? And if we look at the first function, the AUC for the first function is 80.8, for the second function is 0.767. And for the third function, we cannot compute AUC because we don't have zeros. So we only have ones. Does it make sense? And that's a big problem right now because most of the annotations that we have present on these trees are ones. So it's very biased. Okay, but we're working on that. Okay, so regarding this, before jumping to the future research, so the key takeaways are, so we have a parsimonious model, very simple model for predicting gene functions. It's in phylogenetics. It's computationally scalable. In Sifter, it will take about 66 years to fit the model. For us, it's like 5 minutes. In the case of the results, this is another thing that is meaningful. We get biological meaningful results. In the paper of Sifter, they actually fit the model once. But whatever they get is actually, it doesn't make much sense. So what they do ultimately is just they fix some parameter values that are biologically like make sense, and then use those for making predictions. In our case, we actually are getting our parameter estimates from the model, from the data itself. And regarding the accuracy results, it's comparable to what we observe in other state-of-the-art phyllo-based models. Regarding the future, and this is something that makes me that I'm more excited about. So first, we have been thinking about how can we scale this in a more proper way, because when we pull the data, we just pull the data and treat them all independently. But we didn't. A nicer way of doing this would be, for example, try to do a hierarchical model, which we assume that we have a distribution of parameters, and then for each tree we got the parameters of that tree. Well, we're drawn from these parameters, so we can do something like that. But that might be a bit hard to justify to fit. So how many classes of parameters we would pick, or so that's something that we're not very sure about. But this is another thing that is actually I'm very interested and keen to try it out, is using a framework similar to exponential random graph models. We will be talking about exponential random graph models in March with Professor Mandram. But for now, just let me give you like the fundamentals for you to just get an idea. So in an ergon, exponential random graph models, what we're trying to model is essentially an array, a binary array. So we have an array with zeros and ones, and we're trying to see how can we better describe this array using sufficient statistics, and that's the key. Okay. So let me show you an example. So suppose that at each point in evolution, when we're doing the transition from parent to offspring, we have something like this. For example, here we have the parent, okay? And in which we have the root now. Imagine it has these annotations. The functions A, B, C. It doesn't have a function. It has the B function. It doesn't have the C function. Okay. And then we have two cases. Imagine that these are two offsprings. Using sufficient statistics, it will be something like this. So for example, we can try to count, okay, so how many gains do we observe from parent to offspring? In the first case, we observe just a single gain, right? So, the columns are the offsprings, and these are just two different cases. So in this first case, only one gain. So the second offspring gain a new function, function C. In this other case, the first offspring actually gained two functions. Okay, so the sufficient statistic for this will be 1 and 2 respectively for these two cases. How about how many times we only see one of the offspring changes? So you see in evolution, you sometimes may have like, I don't know, multiple offsprings. And as I was describing earlier, when a change happens, it usually happens in only one of the offsprings. So we can use a sufficient statistic that actually tries to capture exactly that. In this case, only the second offspring changed. The first offspring stayed the same, right? It has the same set of annotations of the parent. But in the second, actually, let me see. In the second, actually, this is actually a binary. I think is a 0 because actually both of them change. Okay. And we can also count swaps. So actually, this more than swaps. I have to remember to rename this. How many changes overall we observe? So how many times we observe that we switch from a 0 to 1 or from a 1 to a 0? Okay. And in this case, we only see 2, right? The second offspring lost the B function and gained C. But then in this case, the first offspring actually swapped all 3 functions, and the second offspring lost the second function. So we observe a total of 4 changes. Okay.
Okay. Why did you go with swaps and just not number of losses?
You can do that as well.
Okay. It just seemed more logical.
Yeah. No, so you can count how many losses, or for example, you can do like a pairwise comparison. So for example, how many times you observe 2 functions being gained together. Because it could be the case that biologists believe that functions A and B are somehow associated. So for your sufficient statistic count, we can use how many times these 2 functions are either gained together or lost together, right? But the key point of all of this is that we can actually use this to build a reduced form of a model, not trying to do the full enumeration of the Markov transition matrix. Because in Sifter, in which they actually use the Markov transition matrix to, for example, to build a model which has only 3 functions, okay, that may interact with each other. For just 3 functions, we need 64 parameters in Sifter. Imagine if we're dealing with 3 functions plus trying to have interactions between the siblings. So the number of parameters here grows to 2 to the 2n. So that it's highly complicated. But in our case, actually, we can try to do all of these calculations, but instead of looking at the whole transition matrix, we just look at the sufficient statistics. And that's the key point. And because of that, we can actually build very complex models in which we make associations between siblings, and at the same time, we can actually make associations across functions, so on and so forth. Does it make sense? Yes?
Do you assume that 2 parents have the same function here?
If 2 parents have the same function?
Yeah, do you assume the 2 parents have the same function?
No. So in this data, at least in this context, we have a single parent. But you could in principle, right? So because again, so all of this is just trying to model an array, right? So what I'm showing you here is that we're trying to calculate this probability based on the state of the parent and the offsprings. But if this is just arrays, so as you are saying, if you are thinking about having, I don't know, if you have 2 parents, this can actually be possible with 2 parents. Because instead of a single vector, you will have 2 vectors, right? So you will have a parent 1 and parent 2. And the functions here, right? And then you have the offsprings. So essentially, this is just you can stack it together, and again, it's so at the end of the day, it's just an array, a binary array. And because it's a binary array, we can do whatever type of counts we want. Does it make sense?
In this graph, I think the parent represents the species.
Yes.
So it's not 2
yeah, you want to have like a, yeah, in genes, I don't see how can you have two genes reproducing. I don't know much biology, it doesn't make sense to me, but yeah, but in principle, the bottom line of this is that we're just modeling these arrays, and whatever we want to like count on this, we can do it using sufficient statistics, and that simplifies the model significantly. And again, so right now the nice thing of this is that in the current version of the model, we are treating everything independent. Functions are independent, series are independent. But if we look them as stacks of data, as arrays, we can actually use sufficient statistics to treat them together. Okay, and that's it. Thank you. Any question?